This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. From Microbe TV, this is TWiM, This Week in Microbiology. Episode 133, recorded on August 16th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. This episode is also brought to you by Drobo, a family of safe, expandable, yet simple to use storage arrays. Drobos are designed to protect your important data forever. Visit drobo.com to learn more. Listeners can save $100 on a Drobo system at drobostore.com by using the discount code microbe100. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there.
Hello again. How have you been?
How are you doing?
Have you been well?
Yes. Traveling too much, but doing fine.
How can you travel too much? The world is an interesting place, no?
It is, but 5 trips in one summer for an old man wears me down.
Well, I hope you stick tight for a while.
I'll try.
There's plenty in San Diego to keep you amused, right?
You're not kidding. You're not kidding. You don't have to go any place for.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Have you been having the same heat wave that we have?
Oh, yes. Every morning, it's like going out into a swamp.
Yeah, it's amazing.
The humidity is very high, and I heard on NPR why it's called the dog days of summer. It's because of the stars that move into the sky when the sun is rising, but it really feels hot and sweaty out there. And
why dog? Where does dog come from?
From the star Sirius.
Oh, the Sirius. That's the dog?
Yeah, the dog
okay.
star. The dog star.
I didn't know that.
And it comes up in the end of July and early August.
Mm-hmm.
And that's why the first part of August, it was always referred to as the dog days of summer.
Interesting. All
yes.
right. You just learn everything on a podcast, you know?
And I learned it from the radio.
You learn it, you pass it on. That's okay.
Yes, I pass it on.
All right. It's the 3 of us today. Michelle's on vacation. Enjoying
That's
herself.
good for Michelle.
It is. She's up in Maine, I think. So enjoy yourself. And today we have a snippet and a paper for your late summer enjoyment. Right? Elio's going to treat us with a snippet. Yes, Michael, what were you going to say?
I was going to say it really is. Both papers are absolutely fascinating.
Yeah, looking forward. Elio, are you going to talk about a virus?
I'm going to talk about the virus, but I'm going to talk about the technique.
Yep.
The technique is cryo-electron tomography. I'll explain it in a minute. And what this does is it allows you to look at cells or particles like viruses which are unfixed. They have not been treated with any chemical and in a state which is as close to natural as is possible. I'll explain how this is done. The upshot is that with this, what this technique does for you is it allows a resolution which is stunning. In the case of viruses, 2.8 angstroms, which is atomic level. 1 angstrom being about as small as an atom gets. So this is phenomenal, and it does compete almost, or competes in some cases very well, with X-ray crystallography. So for those of you who are interested in 3D structures, this is where there is a huge development, which is an alternative to X-ray crystallography. So let me explain a little bit what it is. Both the cryo and the tomography are relevant. The cryo refers to the fact that whatever object you're dealing with is plunged at high speed into liquid ethane or something at minus 200 whatever degrees, extraordinarily low temperature, done extremely fast. And what this does is, instead of crystallizing the water, it makes it into a vitreous, glass-like material, which is essentially harmless. And the reason I say harmless is because if you did this with bacteria and you plunge them at this low temperature, you take them out and you throw them, they swim away. They're perfectly alive. So there is no artifact of fixation. The only artifact that exists, and it's very hard to deal with, is the fact that you have to irradiate the material with electrons because you're using an electron microscope. And that, of course, is potentially damaging. So, of course, you go get around that as best you can by using as little of a beam of electrons as you can. However, this brings me to the second part, which is the tomography, and which requires a number of exposures. That is, it's not a single picture you're taking. So tomography means that you're taking optical slices, in this case electron microscopic slices of the object, in the same way that a CAT scan takes slices of your chest. And you can reconstruct these slices into a 3D object, and you can look at each slice singly, or you can reconstruct it into the whole thing. This technique is done by tricks which are a little bit peculiar to the electron microscope. Instead of having horizontal sections, you take pictures at an angle, a series of angles, and so this gives you a slice essentially, and the computer reconstructs this. Now, this technology is phenomenal, and it has led to developments which are totally unimaginable. You can see living cells under the electron microscope, and living until you bombard them with electrons, and you can determine a lot about their structure. It's been used a lot for bacteria, for other cells. There are several limitations. One of them is the object has to be very thin. So either you use very small bacteria, very thin bacteria, or you abrade away slices with an ion beam. You abrade away part of the tissue until you're left with a very thin layer, which you specify where in the cell is the layer. So you can really look at thick objects as well. You just make them thin by planing away. It's like a plane in your wood shop. You plane away layers until you are left with a very thin layer. Okay. With viruses, this has had a particularly big impact because it has been combined with a lot of very fancy computer power. Here's the story. When you deposit viruses, purified virus particles, on an electron microscope grid, of course, they'll fall down in different orientations. Some will be heads up if there is a head. Others will be sideways and so forth. So you can't just look that alone. But what you do is you tell the computer to straighten them out. That is, to reduce all this information to a single particle. And you can do this, and you can do it very successfully, as I say, with a resolution which is stunning. And here's the interesting part. You can do this without having to crystallize the viruses. Obviously, for X-ray crystallography, you need crystals. Well, crystallizing viruses is not always simple. In fact, in some cases, it's rather difficult. It takes a lot of material. It takes a lot of patience. And the tricks of making crystals are sort of an arcana. It's sort of like alchemy in a way. You have to really know what you're doing. However, this technique allows you to look at rather sparse populations. That is, you don't need a lot of virus to see them. And in this case, we're talking here about a paper by a number of authors from Purdue University and University of Wisconsin. The names are Liu, Hill, Close, Chen, Waters, Bochkow, Jiang, Palmenberg, and Rossman, published in the PNAS August 9th. The title is Atomic Structure of a Rhinovirus C, a virus species linked to severe childhood asthma. And the title tells you what this is. So they study a virus which is very difficult to cultivate. And you will not get enough material to make crystals out of it. However, you can grow it and get a few virus particles essentially out of it. You can concentrate them through a sucrose gradient and you have enough material. So the virus is almost a new group of viruses. You want to say something about that?
Sure.
This is your field.
Yeah, in fact, we do work on rhinoviruses in this lab, and we do work on this particular species, the Rhino C. So up until 10 years ago, there were 100 or 150 different what we called serotypes of rhinoviruses that had been discovered, and mostly they were discovered by taking people with common colds, which is a mild respiratory infection, and you take a little bit of their respiratory secretion, you put it in cell culture, and you isolate a virus. But it turns out that there are many more rhinoviruses out there that we couldn't culture, and so we missed them. And until we started doing direct sequencing of respiratory samples, we didn't get them. And so a number of groups not too long ago found by sequencing respiratory samples from children with very serious lower respiratory tract infections, they revealed this new group of rhinoviruses, which are called the rhinovirus C species. And there are quite a few of them. And now we have well over 150 different rhinoviruses altogether. So the idea is that they seem to be associated with more severe lower tract disease.
Not just a common cold.
Not just a common cold, but they can also cause a common cold as well. And then now it turns out that some of the other rhinoviruses can also cause more serious disease as well. But these have been notoriously hard to grow in culture, and that's why this cryo-EM structure is really very interesting.
Mm-hmm. So I should say that this technique is particularly suitable for non-envelope viruses, although you can use it with envelope viruses to some extent. You don't get nearly the same resolution. And with the non-envelope viruses, it's the icosahedral ones, including picorna viruses and flaviviruses, to which rhinoviruses and rhinovirus is picorna virus, isn't it?
Yeah, sure is.
And Zika is part of the flaviviruses, and so is Ebola. These viruses are terrific for this technology. So you don't need to have to grow huge amounts. You can do it, and you get a lot of information. And it's rather quick if you know what you're doing. So there are many wrinkles to cryotomography. You need a special electron microscope with a lot of intensity. So it's not something you can do on your regular microscope, I don't think. I think it takes something very special. Anyhow, so they get their rhinovirus C15A is the name of the strain that they use. In the paper, there are these gorgeous pictures of it, 3D pictures of the virus, and they include also studies of empty viral particles. When you make a virus preparation, often you end up with empty virus capsids. They don't have the nucleic acid in them. And it turns out that they have the same structure as the whole particle. And this is of some importance because they could be used in vaccines. It seems clear that, the capsid is the immunogenic part of the virus. You don't need the nucleic acid, and not having the nucleic acid, of course, makes the particles non-infectious, non-dangerous. So a capsid-based vaccine is probably a good idea. You want to say something about that, Vincent?
Yeah, for many picorna viruses like polio, the empty capsids are different or antigenically different from the capsids with RNA in them. But here, the structures show that at least on the surface, they look very similar. So you can imagine that you could use an empty rhinovirus C particle as an immunogen. And of course, that has to be tested and so forth. But these rhino Cs have a very unique structure among the rhinoviruses. They have these little fingers sticking out on the surface. And that was found in both the empties and in the virus with RNA in it. So it's interesting.
But more importantly, I think what this technique highlights is that it will be much quicker to develop a vaccine candidate that you could either test in an animal model or test even in people
Right.
because you know what it looks like to our
Right.
immune system.
Yeah.
And here they in fact go into some detail about a canyon, which is a pocket at the base of protruding spikes on the surface of the capsid, and these are probably the place where they interact with a sialic acid-containing receptor. They derive this in part from studying an enterovirus, but it looks very good that they can really pinpoint the place which matters in the infection. I think this is really a nice, sophisticated study. And by the way, the paper is accompanied by a commentary in PNAS called Cryo-EM of Viruses and Vaccine Design by Earl and Subramanian. And I must say, I was caught by this title, Cryo-EM of Viruses and Vaccine Design. You would think that there would be a big leap there, but there isn't. And so just like Michael just said, this is potentially a very useful way to dissect a virus and determine which candidate is suitable for making a vaccine. It's good stuff.
So this comes from Michael Rossman's group at Purdue University, and he has been a powerhouse of virus structure. He's been in it for many, many years. In fact, he developed some of the earliest computer algorithms for determining structures, and that was back when you had to do X-ray crystallography to get virus structure. And now he's been ripping off the virus structures. Last summer, he did enterovirus 68 within a few months of the outbreak. Then earlier this year, he did Zika virus, and now he has rhinovirus C. He's really got an amazing group. And I should point out that he says in this paper that all the primary data on which the structures were created were collected in less than 1 week. That's amazing.
It really is.
Collecting data in a week is amazing because in the old days when you had crystals, you had to go to a high-energy synchrotron. You'd spend months and months and months collecting data. Look at that, less than a week. This is fabulous. This is like, high-throughput sequencing where you can do everything quickly.
But I think a lot of this has to do with the advances in photography that have been made possible by everybody having a camera in their pocket. Those of you who understand your iPhone and understand how it takes pictures, it often takes pictures in a burst mode using different focal lengths, which are the equivalent of different angles, if you will, as this technique is doing in order to get the best focus and best exposure of your particular subject. So a lot of this software that has been made possible to enable us to do this for advancing vaccine design has really been pioneered by the advances in medical imagery, principally CAT scans and MRI tomography. That
also from astronomy. Some of these cameras were developed for astronomic purposes. So,
and I think that's
cross-fertilization.
And that's what really needs to be stressed as to the general value of basic research, is that it's just not one discipline that is advancing this technology. It's many disciplines that are contributing, and then it's the clever people that are taking advantage of advances in other disciplines that really make. If you think about it, we may get a vaccine for Zika very, very quickly because of the phenomenal pace at which structure was designed.
Good point.
I spoke with Michael Rossman, I don't know, about 2 years ago. I did a series of interviews for our textbook, our ASM textbook, Principles of Virology. And if you own the textbook, you can find a link to his interview in the structure chapter. But during that interview, I asked him, we were talking about how computational power has changed over the years. And I said, what kind of computer do you use today? And he pulled out his iPhone.
Oh, my God.
Yeah.
Oh, my God.
It really is. You truly do have a supercomputer in your pocket.
Amazing.
Yeah. Anyhow, the uses of cryotomography extend themselves way beyond viruses, where obviously they have already had a huge impact. So everybody should tune in on that because it's part of the armamentarium if you allow a long word
Yep.
of modern biology. It's
bet.
really part and parcel of what you do. And in time, I think the technique will be, the machine that does it will be simplified and made more available. Clearly, these are the early days in terms of some of the technology.
You bet.
It's going to be like advanced sequencing. I mean, before you had to have that big box. Now a lot of folks are, getting their machine on their desktops.
Yep. Right. Thank you, Elio.
Of course.
I want to, before we move on to our paper, I want to tell you about our first sponsor, CuriosityStream. They're the world's first ad-free nonfiction streaming service founded by John Hendricks, the founder of Discovery Communications. So you know you're going to get science, not reality TV. They have over 1,500 titles, 600 hours of content available in 196 countries, and you can get them in many ways on the web and with any of these devices for streaming video: Roku, Android, iOS, Chromecast, Amazon Fire and Kindle, Apple TV. And what they have is a wide variety of science and technology content. So, for example, I'm on their site right now, and if you go to browse, you can pick science, technology, civilization, human spirit, artificial intelligence, communications, energy, engineering, nanotechnology, and so forth. So if I pick science, I come up with The Secrets of Quantum Physics, Exploring Quantum History with Brian Greene, who's a professor here at Columbia, Deep Time History, Parallel Universes, Viruses, Destruction and Creation, Jason Silva, Transhumanism, The Rise and Fall of T-Rex, The Hawking Paradox. So you can see lots of, Are Genes Under Influence? Lots of great stuff. So if you love listening to TWiM and the other podcasts that we do, you might like this. You can check it out. Plans, there are monthly and annual plans available, and they start at just $2.99 a month, less than a cup of coffee or the cost of one title on another platform. I'll tell you in a moment how you can check it out without paying anything. You go to curiositystream.com/microbe, use the promo code microbe during sign-up, and you'll get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 entire months free of one of the largest 4K libraries around. Go to curiositystream.com/microbe and use the offer code microbe at sign up, not startup. So you can, I always say startup, I don't know why, but you can check it out for 2 months. If you don't like it, it's the end of it. But I'll bet you like a lot of these programs. And we do thank CuriosityStream for their support of TWiM. And now we have an interesting paper that Michael wanted to do a couple of weeks ago, but we deferred him, and now it's his turn.
It's now my turn. This is a paper that came out in Nature at the end of July, and it's entitled Human Commensals Producing a Novel Antibiotic Impair Pathogen Colonization. And in this group out of Tübingen, Germany, out of Andreas Peskel's laboratory, and the first authors, there's actually 2 first authors, Alexander Zippier and Martin Konerath. And it's also got co-authors of Claudia Laux, Anne Burscheid, Daniela Janik.
You're sure you want to go through that? There are 17 of them.
There's 17 of them.
Why don't we just skip that?
We'll just skip that. And I mean, no disrespect to this phenomenal group of authors. And it's really a neat paper. The study that we're going to take you through here is shows that Staphylococcus lundinensis, which is a commensal bacterium that shares the nasal niche with Staph aureus, is associated with a reduced ability of Staph aureus to colonize the human nose. Staph lundinensis is a coag-negative staphylococci, and in generally speaking, coag-negative staph.
Spell out coag.
It's coagulase, and so it depends on how you abbreviate things. It's COAG is generally the way I shorthanded in my notes, but it stands for coagulase, which is an enzyme that coagulates rabbit plasma, and it's an enzyme that the Staph aureus has, and the pathogenic variants of Staph aureus are typically coagulase positive or coag positive. and it's one of the virulence determinants of Staphylococcus aureus. Lundinensis is coag negative or coagulase free, or it lacks that particular enzyme. And the topic of this paper, as the title implies, is the description of a new antibiotic that was literally synthesized in our own noses, and it's synthesized in situ. It's a secondary metabolite, which means that it's generally made as the organism goes into maximum stationary phase. It's just as the organism is going to bed, if you will, metabolically, that it synthesizes this peptide antibiotic that is not synthesized by our ribosome, but is actually non-ribosomally manufactured by the bacterium. And we'll get into the meat of that here in a few minutes. So some background before we get into the meat of the paper. 1 in 3 of us have been colonized by Staph aureus in our noses. Staphylococcus aureus is generally considered a pathogen when it moves out of our warm and moist niche that is our nose, or our nasal cavity. And it generally results in death and destruction when it wanders away from the nasal pharynx. 2 out of 10 of us, unfortunately, have the drug-resistant variant of Staph aureus, or MRSA, which stands for methicillin-resistant Staphylococcus aureus. And MRSA kills about 11,000 individuals in the United States each year, and it's the most likely culprit for a hospital-associated infection that results in death should you acquire this pathogen while you're being treated in an intensive care unit while you're hospitalized. And MRSA, along with vancomycin-resistant enterococci and the gram-negative organisms that are becoming resistant to the third-generation cephalosporins, are, of course, the big 3, not the automakers, but the big 3 antibiotic-resistant strains that everybody is very much concerned about with hospital-associated infections. And so there are 5 parts of our story today, and I'm just going to list them out for us. The first is the antimicrobial activity of the nasal commensal, Staph lundinensis. The second is that this microbe, Lundinensis, produces and secretes lugdunin, which is this novel non-ribosomally synthesized peptide antibiotic. The third area is that this novel peptide antibiotic is quite remarkable, and it has a broad spectrum of activity at a very low concentration, typically micromolar concentrations, and it's active against a wide variety of the major human pathogens. The fourth, and this is where it really gets into the interesting aspects of the microbiology, is lugdunin production facilitates Lundinensis' ability to compete in the nasal niche to actually prevent Staph aureus from coming in and taking over. And the experiments that they use to demonstrate this aspect is pretty neat when you see how they did the experiments. And finally, the fifth aspect is the interference with Staph aureus carriage. And they did this in a clinical population. So they actually screened 187 patients from their hospital and asked the question, do they have Lundinensis? Do they have Staph aureus? And they learned that the Lundinensis actually, when it was making this peptide antibiotic, was able to keep the evil Staph aureus from colonizing the human nose. And so they really bring together the discipline of microbiology in a neat way. And this paper has something for everyone. So as we begin, we're going to start with step 1, showing the antimicrobial activity of this nasal commensal. And the authors point to us how they began to characterize the staphylococcal isolates associated with our nose. And there's a paper which they call your attention to, reference number 25, which is in one of the PLOS journals. So it's open access. So you'll be able to see how they screen for antimicrobial activity from the isolates from the human nose against Staph aureus. And that's effectively how they found this particular strain of Staph aureus, or excuse me, Staph Lundinensis, that was producing this antibiotic. And this antibiotic is only produced under conditions where it's iron limiting, and it's as the cell is going into maximum stationary phase or secondary metabolites. And so the way they did the experiment—
That's not atypical, right?
It's not typical. It's not typical. And it only produces this peptide antibiotic on a solid surface. So traditionally, the way you screen for antibiotics is you isolate some fraction from the culture liquor. You either extract it with a solvent, or you ask the question, is it water soluble? And you deposit it on a filter paper disc, and you look for a zone of inhibition, a la Kirby-Bauer. So this is not your father's Kirby-Bauer test. This is where you put Lundinensis on the Petri plate, and it actually forms a colony, but you deposit it on top of a lawn of Staph aureus. And the Staph aureus that they used was USA 300, which is the pathogen. Go ahead, Elio.
Tell me, is this, do they, I've forgotten, do they tell the story of how they found this unusual way of detecting an antibiotic?
That's reference 25, which is in the PLOS journal, and it's buried on my desk.
Do they say how they found it? Is it they found that some colonies were inhibiting other bacteria? Is that?
Yes, they were specifically looking for inhibition of Staph aureus USA 300.
But like you say, this is not the usual way to look for inhibition. So they must have had a mixed culture with Lundinensis inhibiting the growth of something else, of Staph aureus. So they must have looked at the colonies on the plate. So it's a very different way of looking at it.
It is indeed a very different way of doing it, which is why I'm calling everyone's attention to reference 25, which is in the PLOS journal, PLOS pathogens. And it's nutrient limitations govern Staph aureus metabolism and niche adaptation in the human nose. And it's in PLOS pathogens, volume 10 from 2014. And there's a specific reference. So it's by Krismer and colleagues. And so that's effectively how they were characterizing it. So what they then did is they began to characterize the strain of Lundinensis that was producing this novel antibiotic, and they conducted Transposon Mutagenesis, and they were able to identify the location within the chromosome. And what they learned, it was about a 30 kilodalton insertion that was, 30 kilobyte insertion of
kilobase.
Yes, I'm just having the PLOS pathogen screwed me up here. They characterized the operon, and they learned that it encoded four genes that were responsible for the synthesis of this non-ribosomal peptide. And it's an interesting molecule because it's a heptapeptide, and they then take us through the characterization of this gene cluster and the proposed biosynthetic pathway of lugdunin. And it's rather interesting in how they began to characterize it. And they used reverse phase high pressure liquid chromatography coupled with ultraviolet and mass spectroscopy. And they were able to reveal obvious differences between the wild type lugdunin producing Lundinensis and the transposon mediated mutant, which had a particular knockout for one of the genes. And without taking you through the discussion of this, they then cloned out this 30 kilobase operon and were able to put it under xylose sensitive control, and they would add the sugar and they would be able to overproduce the lugdunin, and they were then able to extract it using alcohol so that they could effectively get enough of this material from which they could further characterize this particular cyclic antimicrobial, which is comprised of a thiazolidine ring structure that, and the 5 amino acids in this are rather curious. It's a mixture of both D and L amino acids.
It is.
So they have
interesting.
So they have D-valine and then L-tryptophan and D-leucine and L-valine and D-valine, and it forms this beautiful cyclic thiazolidine that is got this profound antimicrobial activity. And this lugdunin is rather remarkable in that it has a wide range of antimicrobial activity against a lot of these difficult to treat things like methicillin-resistant Staph aureus. And when they began to further characterize it, they found that this cyclic peptide antibiotic was remarkable in that when they added it to cultures, it immediately stopped the production of DNA, RNA, protein, and cell wall precursors. It literally hit the major targets of all antibiotics. Traditionally, an antibiotic inhibits one process. For example, tetracycline inhibits protein synthesis. Penicillin inhibits cell wall formation. Chloramphenicol, protein synthesis. This one knocks out everything. And they did this via the addition of radioactive precursors, and they asked what it knocked out. And there's only one other antibiotic that I know of that has got such broad spectrum ability to literally crash and burn the entire cell system, and that's, of course, daptomycin. And no one really quite understands how daptomycin works. And when I was reading this, it immediately suggested to me what this antibiotic is likely doing is collapsing the ability of the cell to deal with its energy, because it literally is crashing everything. And it works very similar to something that I focus on a lot is how does copper kill bacteria so quickly? And it literally collapses everything. And so they're still working on the mechanism of how it works. So they then asked this next question, whether or not lugdunin could arrest an infection in vivo. They did this with a mouse skin model that was reflective of what Staph aureus does in people. Namely, it causes abscess. And in the mouse model, what they do is they shave a mouse, and then they put skin. They wound the skin by stripping it with adhesive tape, and then they add Staph aureus to that. And then they measure at 24, 30, and 42 hours post inoculation of the Staph aureus to this wound, they add 1.5 micrograms of purified lugdunin, and they allow it to sit for 6 hours, and then they ask, did the infection go away? And they did this by measuring the concentration of bacteria. And the control they show, the bacteria continue to grow, and they get to an extremely high concentration, upwards to 10 to the 10 per mil of tissue that they analyzed, while within 30 hours of the addition of lugdunin to these skin punch biopsies, all the bacteria or Staph aureus that they added not only did not grow, they're all gone. They did another really neat experiment, is they cultivated Staph aureus, or they did successive cultures of Staph aureus in sublethal concentrations of rifampin. And rifampin inhibits RNA synthesis. And if you grow Staph aureus in the presence of rifampicin, it quickly becomes resistant to it. And so in this particular experiment, they're able to show that indeed the Staph aureus does become resistant to rifampicin. But when they did the same sort of experiment with Lundinen, they showed that the Staph aureus was not able to overcome. So there was no selective advantage. There was no ability to select against this particular antibiotic. And I think it's principally because it's so fundamental in how quickly and completely it can shut down the cell's ability to live. It literally just collapses everything. And so they're hopeful that Staph aureus won't become resistant, as well as other microbes won't become resistant to Lundinen. And then they asked the question whether or not it would actually protect animals. And again, they have this wounded black 6 animal that they've added the Staph aureus to, and they asked whether Lundinen could actually eliminate the bacteria both at the surface and then deep within the tissue. So they effectively count the bacteria at the surface of the wounded skin and then deep in the skin biopsy. And they were able to show in these mice that Lundinen not only was able to literally collapse the ability of Staph aureus to cause an infection in these animals, but it was able to actually penetrate deeply into the tissue, effectively down the hair shaft, to prevent the Staphylococcus from really beginning to set up housekeeping. And again, remember, Staph is really good at attaching itself to us, and then again becoming locally invasive, which is one of the hallmarks of a staphylococcal infection. They also bring to our attention the curious behavior of mice. And if you look at their data, you see there are 2 mice in the cohort that didn't seem to respond to Lundinen. And what they report is the mice have this incredible ability to lick their wounds, and consequently they think that what happened is the mouse was licking the Lundinen off of its back before it had the ability to do its activity.
They're going to put the collar around the mice just like they do with dogs?
I think that's what they're going to have to do. But, it shows that they're really confident in this antimicrobial at being able to stop these infections cold in its track. And this takes us to part 4 of their study, the why. And this takes us back to thinking about the microbiology of why organisms would carry a 30 kilobase region of extra DNA. And they comment in their characterization of the of this gene array or operon, they actually reported a GC difference between this operon. It was 26.9% G plus C, while the whole genome of Lundinensis was 33.8% G plus C. And this is reminiscent of a pathogenicity island that has likely brought Lundinen into Lundinensis. And they further comment in their characterization of this operon that of the published DNA sequences, they found that this Lundinen operon was actually present in everything that they looked for. So this is really arguing that the horizontal gene transfer from one bacterial species to another is actually conferred a selective advantage to Lundinensis because it's carrying it in everything that we have sequenced that's out in our database. So it must confer some fitness to this particular strain. And so this then takes us to the next suite of experiments that they're asking about, namely whether or not Lundinen producing wild-type Lundinensis will restrict the growth of Staph aureus first in vitro and then in vivo in cotton rats. And just an aside here, I worked with cotton rats when I was a first-year graduate student to supplement my income, and these are the meanest, nastiest rodents you ever want to deal with.
Did you get bitten? Did they bite you?
No, I wore gloves, and they were leather gloves. But they are mean and nasty little creatures. So they have a knockout in which they knocked out by gene replacement design the LugD, which is the smallest portion of this 4-gene operon. They knocked out LugD with gene replacement. And what they show in the first experiment is they take in a ratio of 90 parts Staph aureus to 10 parts Lundinensis, and they ask the question whether or not the wild-type strain that has Lundinen versus the mutant strain that has got the deletion, whether or not Lundinensis would be able to suppress Staph aureus. So they add these first in vitro, and they measure the ratio of the bacteria by percent, and what they show is that the wild type producing Lundinen is able to quickly displace Staph aureus, and it goes lickety-split from within 2 days of incubation or 48 hours, virtually all the Staph aureus is gone, while the mutant that's been deleted for LugD, there's really no difference. The Staph aureus is able to compete quite well, and poor Lundinensis is unable to compete. And then they have a complementary plasmid that they add back to the system, and they're able to show that it indeed does complement the ability to compete. So they need LugD in order to make an intact Lundinensis or a Lundinen antimicrobial to enable the competition. So Lundinen producing strains of Lundinensis are able to outcompete Staph aureus. And then they did this in these mean, nasty cotton rats, and they have the wild type variant and then the mutant variant, and they ask when they inoculate the nose of this cotton rat what's present in the Staph aureus. They're able to recover about 8,000 colony form units per nose of cotton rat, while Lundinensis is able to significantly reduce the concentration, or the wild type is able to significantly reduce the concentration of.
This sounds like a really magic drug. Now, about the resistance part, we've heard this before, right? Almost every time a new antibiotic comes up, people say, oh, we don't find resistance to it. So we have to take it with a grain of salt, don't you think?
Yeah, I do indeed think we have to take this with a grain of salt. But I think because it's so profound in its ability to shut down the 4 essential processes, DNA, RNA, protein, and cell wall biogenesis, I think that's going to show us that it's going to take longer for organisms to evolve unless
I don't know. Unless it's an
plasma.
uptake system. I mean, all they have to do is to exclude it or to spit it out by a secretion mechanism. That doesn't sound so impossible.
No,
the fact that it has many targets doesn't matter. If it doesn't get in, it doesn't get in.
But what argues against it is the final set of experiments in this particular paper, and that's Staph aureus and Lundinensis distribution in hospitalized patients. And here they make the argument against this resistance pattern, and they make also the argument that because Lundinensis seems to, all the strains of Lundinensis that have been sequenced and isolated seem to have this operon. It's really arguing that there's significant difference between Lundinensis positive and Lundinensis negative patients associated.
That may be, but this is if the drug went on the market and were used in large amounts, this is a totally different situation than having it made in a relatively small amount by the native bug. So I'm not sure that this doesn't sound like it's a tight argument.
Well, it obviously needs more animal studies to see whether or not animals will be able to develop resistance, and then whether or not it will survive safety trials, and then whether or not it warrants a full-scale clinical trial.
It certainly sounds like a very interesting drug, and it has some characteristics which are very different than others. It's really interesting. Nice work.
Yeah.
So that's the paper.
It's very nice. It's cool stuff. So this has some promise, but they have to go through preclinical and phase 1, et cetera, right? But do you think that will happen?
There's no telling. We desperately need new drugs against Staph aureus.
Mm-hmm.
Remember, daptomycin was isolated back in the late 70s, and it only came to market in this century.
Yeah,
that's right.
So
takes a long time.
Yeah, the fact that they've published it maybe mean that they're not even going to pursue it. My old friend Lynn Enquist used to always say, if you see them publishing an antibiotic, it means they're not going to go forward with it. But we'll see.
You never know.
You never know. Before we move on, I want to tell you about our other sponsor of this episode, Drobo, which makes devices for protecting your data. They have systems. These are black boxes, and you put drives into them that connect directly to your Windows machine or your Mac via USB, Thunderbolt, and they also have network storage devices. And now an interesting thing, we've told you a little bit last time about Drobo and how they work. Now I want to tell you that they decided to encourage developers to develop Drobo apps. So they made the correct software available to them to let them do this. And they're hoping that developers will make apps that allow you to do different things, like back up your device to different cloud service providers, get remote access to your files on your network-attached Drobo, develop websites on your Drobo, entertainment, stream your media to computers using software like Plex, or distribute your media with tools like a BitTorrent client for gathering files, or even email. You could run an email server on the Drobo using the software that's written. The models 5N and 810N are network storage systems, which can be extended by Drobo Apps to do a variety of different things. And point here is that the data is encrypted end-to-end, ensuring your privacy. We'll tell you a little bit next time about how MyDrobo and Drobo Access work to allow you to set up your Drobo, your network-attached Drobo, say at home or at work, and then you can get your data elsewhere securely. And it's really very, very interesting and exciting. I have one that I put in my lab, and I want to share our lab data among the people in the lab, but we'd also like to be able to access it from outside. And that's always been challenging here at Columbia. We'll see how it works, and I'll fill you in on that. So Drobo is our sponsor here on TWiM, and we would like to tell you that you could save $100 off on the purchase of a Drobo Mini, 5D, 5N, or any 8 or 12 drive system. You go to drobostore.com and use the discount code microbe100, and you get $100 off on your choice of those systems. We thank Drobo for their support of TWiM. We have a couple of emails, 2 emails here to give you. One of them is from Arthur, who writes, "Hello, Twimmers. I'm a graduate student working in mycoplasmology, and I must say I've been overjoyed to hear the past few episodes mention our tiny friends, mycoplasma. Elio's mention of their unique mechanism of moving, gliding motility made me think of a recent paper from the group in Japan under Dr. Miyata. You may find the paper quite interesting. They've done a lot of work on mycoplasma motility, and local U.S. researchers such as Dr. Mitch Baelish at Miami University also have made great advances in the field. In discussion of the MIB system, that's the cleaving system that chops up the antibodies we talked about not too long ago. One thing that has caused some head scratching in our lab has been why the microbe cleaves the FAB portion rather than the FC. The human species M. genitalium and M. pneumoniae don't have the serine protease, at least based on homology searches, and bind to IgG nonspecifically. It makes you wonder why the microbe has conserved that binding protein. Quite a neat area that needs more work. It's a good question. Thanks for
the simple answer is a selective advantage to the organism.
Yeah, right.
Yeah,
could be. Thanks for such an interesting podcast and keeping me preoccupied during my hours in the mouse facility.
Hey.
I sent a link to a paper called Periodicity in Attachment Organelle Revealed by Electron Cryotomography Suggests Conformational Changes in Gliding Mechanisms of Mycoplasma pneumoniae. And it's interesting, Elio, that they use cryo-electron tomography
How about that?
To study the movement, the gliding of mycoplasma. Very interesting. Our last email is from Frank, who writes, Dear Doctors of TWiM, 2 observations on pili conduction. TWiM number 51 featured Hazel Barton discussing her discoveries on microbial excavation of caves. Barton's comments on the role of geobacter electron transport in speleogenesis would be very interesting. Well, yeah, I agree. I emailed Hazel, but she hasn't answered.
She's probably in a cave.
She may be in a cave. Literally.
It is summertime.
Literally. It's cool in the cave, right?
It is.
Elio, do you think that this electron transport would have some role in the formation of caves?
Well, that's a good question. I don't know. I mean, the caves in calcium carbonate caves are made by acid. Sulfuric acid, which comes from H2S, which seeps through, which bacteria oxidize to sulfuric acid. Is electricity involved in that? Well, why not? Could be.
It's how batteries work.
Yeah,
it's how batteries work.
Remember the car battery, the sulfuric acid.
Right. Yeah, that's right. And Frank continues, secondly, biobatteries would seem to be far ahead of their time. These energy sources that are so valuable to low CV biological systems are likely not going to be useful in our crude, relatively high CV electronics. Current and voltage, right?
Yes, current and voltage.
I have to imagine that our current electronic technology will eventually discover and utilize biomolecular processes to accomplish computing and communication. At that time, biobatteries will come into critical use, and we can start to approximate the complexity and elegance of multicellular organisms. August gloom pervades the Northern California coast, where the temperature is UGG 0.7, 15.5 C, and the humidity is only a optical tease of much-needed falling water. Thanks for all the education. Your importance can't be overestimated. Best regards, Frank.
Nice. Thanks, Frank.
Yeah, I do think it's early for biobatteries, but there's some low CV applications where they might be used first. But that's a good point.
Or sensors in the ocean, for instance.
Exactly.
The New York Times did cover a story about using biobatteries, feeding your cell phone a little glucose to feed the bacteria that would charge up your phone. Okay. And it's not without precedent. So it's just a matter of reducing the current need. And as we all know with our cell phones, is the cell phone designers are trying to get our screens to use less juice. And it's only a matter of time until we can, reduce the current and voltage needs.
That's right.
And I think that's going to come with time.
All right. That's it for this episode of TWiM, which you can find at iTunes and also at microbe.tv/TWiM. You can send us your questions and comments to twim@microbe.tv. We love getting them, hearing about you and answering your questions. And consider becoming a patron of TWiM and the Microbe TV family of science shows. Go over to patreon.com/microbe TV to learn more. I received an email from one individual who didn't like having ads on TWiM. And, I don't blame you. I didn't like ads either. And the alternative is to have all of you or some of you support us. You could do that at Patreon. And if we reach our goal of support at Patreon, we'll get rid of the ads. How about that? I'd love to have it a listener-supported show. But I don't know if that would work or not. We do have very nice number of supporters so far at Patreon, but we'd need more to get rid of the ads. So that could be our goal. I'm patient. I will wait. Elio Schaechter is at the wonderful blog, Small Things Considered. Thank you, Elio.
Thank you so much. Appreciate it.
And you should check out his book, In the Company of Microbes, which is an excerpt of posts at Small Things Considered. Go over to ASM Press and find that.
Also, his other book that Michelle's a co-author of, Microbe.
I haven't gotten my copy yet. I'm waiting for that.
Oh,
waiting with bated breath. I got another copy of In the Company of Microbes yesterday in the mail. I have two now. I have one that's autographed and one that's plain. Maybe I'll give it to someone, right?
Okay, yeah, I'll give it to your favorite student.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
And I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Chris Condayan and Ray Ortega for their behind-the-scenes technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.